Hi, good morning, everybody. Welcome back to daily lectures by New Age Cosmetic White, EdTech Healthcare Consultancy. I'm Dr. Bobby, and we're on the subject of molecular biotechnology under the specific chapter of transgenic animals, and we're on to the subtopic of improving animal production traits. While improving production traits, such as muscle mass in meat animals, is often more difficult than engineering animals to express a foreign gene in milk because multiple genes involved in controlling growth and composition and a detailed understanding of the genetic basis for the traits of interest is required. Initially, researchers sought to increase the body mass of livestock by introducing genes encoding growth hormones or insulin-like growth factor, although these early efforts yielded animals with increased ability to convert feed into body weight, they were hampered by difficulties in controlling the expression of the genes, overproduction of growth hormone, which found to adversely affect the animal's health, which manifested as gastric ulceration, kidney dysfunction, lameness, inflammation of the lining of the heart, immobility of the joints, and susceptibility to pneumonia. The reasons for these symptoms are not known. To overcome this problem, inducible promoters were used, such as the metallothionein promoter, which can be activated by zinc administered in the animal's diet. However, expression was often poor because the trans gene was incorporated into heterochromatin, transcriptionally silent regions of the genome. Some cattle breeds, such as Belgian Blue, have larger and leaner muscle mass than other breeds because of natural occurring mutations in the gene encoding myostatin, a growth factor that normally inhibits the growth of skeletal muscle. Interestingly, humans with rare mutations that disrupt the function of myostatin are unusually muscular, and dogs of the whippet breed that have enhanced muscle mass known as double muscling and increased racing speed carry mutations in both copies of the myostatin gene, MSDN. These mutations result in production of a truncated and therefore non-functional myostatin protein. Some researchers have reasoned that a transgenesis approach could be used to disrupt myostatin function in other cattle breeds to increase meat production. Moreover, if disruption could be limited to male animals, then milk production females would be unaffected. The feasibility of this approach has been demonstrated in mice where a myostatin inactivator was targeted to the Y chromosome so that it would be expressed only in male animals. The myostatin inactivator consists of the N-terminal propeptide domain latency associated peptide LAP of the myostatin protein. From proteolytic cleavage, the N-terminal propeptide can hold the C-terminal portion of the protein, which is the biologically active component. In an active state, this blocks myostatin activity that would normally lead to inhibition of muscle growth. A two-step strategy was used to target the gene encoding the myostatin activator, the mouse Y chromosome. In the first step, a cassette containing positive neo and negative HSV-TK selectable markers flanked by Loxp sites were inserted into a non-essential region of the Y chromosome of mouse embryonic stem cells by homologous recombination. Transformed cells were selected by resistance to the antibiotic G418 conferred by the neogene, and the integration site in the Y chromosome was confirmed by PCR. In the second step, a gene encoding the myostatin activator under the control of a strong rat skeletal muscle promoter and enhancer, and also flanked by Loxp sites, was cloned into a plasmid and introduced by electroporation into the transvector embryonic stem cells carrying the selected marker cassette. A second plasmid encoding Cre recombinase was also introduced into the transvected cells. Cre recombinase activity resulted in recombination and the relation between IOXP sites and integration of the myostatin activator gene into the Y chromosome. Exchange of the myostatin inactivator sequence with a selectable marker cassette was initially selected by resistance to Jansekilova, indicating loss of the TK gene, and then confirmed by PCR, transfected embryonic stem cells carrying the myostatin inactivator gene was cultured, inserted into blastocysts, and then implanted into pseudo-pregnant foster mothers. A single transgenic male founder mouse was obtained and then mated with a non-transgenic male females. As expected for a gene integrated into the Y chromosome, all male offsprings carry the myostatin activated transgene, whereas none of the female offspring were transgenic. In the transgenic males, the myostatin activator was expressed in skeletal muscle tissue and not in the heart or liver tissue. Muscle mass was greater by 5 to 20% in transgenic males than in non-transgenic male control mice, suggesting that this may be a feasible approach to increase meat yields in livestock. Larger offspring often lead to birthing difficulties for female animals. Therefore, it may be necessary to use promoters that can be controlled to delay expression of the transgene until after birth. A diet rich in omega-3 fatty acids has been extolled as an aid in the prevention of cancer, autoimmune diseases, such as arthritis and a variety of other diseases. Omega-3 fatty acids are long-chain polyunsaturated fatty acids found mainly in fish. Humans cannot produce these fatty acids, nor can livestock animals, whose tissues are consumed in human diets. Rather, they are acquired from a diet containing fish meal, fish oils, and flaxseed. The tissues of livestock animals contain high yields of omega-6 fatty acids, largely because they are fed a grain diet rich in these fatty acids. Livestock lack the enzymes to convert omega-6 fatty acids to omega-3 fatty acids. Diets with the high omega-6 content relative to omega-3 content contribute to a variety of diseases, including cancer, heart disease, and diabetes. One strategy to increase omega-3 fatty acid content in the human diet is to produce pigs that synthesize omega-3 fatty acids. The Rhomboom Cenorhabditis elegans produces a desaturase that can convert omega-6 fatty acids to omega-3 fatty acids by introducing a double bond to the hydrocarbon. When this gene was transferred to mice, they gained the ability to synthesize omega-3 fatty acids. An optimized version of the FAT1 gene modified codon usage was cloned into an expression vector under the control of the chicken beta-actin promoter and the cytomegalovirus enhancer. It was used to transfect fetal pig fibroblasts. Cultured cells that produce higher levels of omega-3 fatty acids and lower levels of omega-6 fatty acids were used to produce FAT1 transgenic pigs by nuclear transfer. The transgenic pigs showed their threefold higher levels of omega-3 fatty acids and 23% lower levels of omega-6 fatty acids than their non-transgenic counterparts, indicating that they produced the FAT1 desaturase and converted omega-6 to omega-3 fatty acids. Transgenesis is also being used to address certain environmental concerns. For example, a major ecological problem within the massive rearing of pigs and poultry, that is, monogastric organisms, is the overabundance of phosphorus in their fecal material. Phosphorus from pig or poultry manure that is stored outdoors or used as fertilizer can run off into water systems and cause excessive growth of cyanobacterial and algal populations, algal blooms, that in turn deplete the oxygen supply and subsequently kill fish and other aquatic organisms. In addition, large amounts of phosphorus in the environment are implicated in the production of gases that enhance the greenhouse effect and contribute to global warming. Pigs and poultry excrete large amounts of phosphorus because, unlike ruminants, they are able to digest and utilize phytate, myo-inositol 1,2,3,4,5,6, hexakis dehydrogen orthophosphate or phytic acid. The predominant storage form of phosphorus in plant-based animal feeds. For instance, the main food source for pigs is soybean meal, which is about 50% or more of its phosphate of phytate. The inability to catabolize phytate is due to the absence of the enzyme phytase, which is found in plants and microorganisms. Most phytases remove successive phosphates from phytate to produce inositol to monophosphate or inositol. Phytase has been added to animal feed to facilitate the dietary uptake of phosphorus and to lower the phosphorus content of the excreta. However, this supplement is costly and inefficient because much of the enzyme activity is lost during preparation and storage of the feed. Our pigs and poultry excrete large amounts of phosphorus because, unlike ruminants, they are able to digest and utilize phytate, myo-inositol, 1, 2, 3, 4, 5, 6 hexakis hydrogen orthophosphate or pyritic acid. The predominant storage form of phosphorus in plant-based animal feeds, for instance, the main food source for pigs is soybean meal, which has about 50% or more of its phosphate as phytate. The inability to catabolize phytate is due to the absence of the enzyme phytase, which is found in plants and microorganisms. Most phytases remove successive phosphates from phytate to produce inositol, 2-monophosphate or inositol. Phytase has been added to animal feed to facilitate the dietary uptake of phosphorus and to lower the phosphorus content of the excreta. However, this supplement is costly and inefficient because much of the enzyme activity is lost during the preparation and storage of the feed. As an alternative strategy, it was reasoned that transgenic pigs expressed a phytase gene in the salivary glands could overcome the nutritional pollution consequences of phytate in the diet. To this end, pronuclear embryonic macroinjection was used to introduce into pigs a transgene construct consisting of the phytase gene APA from E. coli under the control of the parotid secretory protein promoter that constitutively drives the transcription of a salivary specific protein. In mice, established phytase producing porcine lines were tested for growth in phosphorus excretion using soybean meal with 53% of its total phosphorus as phytate. Under these conditions, the soybean phytate was almost totally digested, and the apical phosphorus content was reduced 75% in comparison to non-transgenic controls. No adverse effects were noted among the phytase transgenic pigs. Tissues from the pigs that are utilized as meat for human consumption contain only trace amounts of the recombinant protein and have essentially the same composition as meat from non-transgenic pigs. Enbrook pig, as it is aptly called, is currently under evaluation by the U.S. Food and Drug Administration for approval of commercialization. On to transgenic poultry. Several features that are unique to avian reproduction and development make the production of transgenic strains by microinjection of DNA into fertilized eggs extremely inefficient. For example, during fertilization of birds, several sperm penetrate the ovum instead of one, as usually occurs in mammals. As a result, it is impossible to identify the male pronucleus that will fuse with the female pronucleus. Also, DNA injected into the cytoplasm of the fertilized egg does not integrate into genomic DNA. Finally, even if nuclear DNA microinjection were practicable, the technique would be difficult to implement because the avian ovum after fertilization becomes a rapid source in rapid succession envelopes in a trough membrane surrounded by large quantities of albumin and enclosed in inner and outer shell membranes. Despite these disadvantages, it is possible to inject a transgene into the regional germinal disc on the yolk that contains the female and male pronuclei. The germinal disc is present before the eggshell is formed. After the administration of DNA to a germinal disc, each egg is cultured in vitro, and when an embryo forms, it is placed in a surrogate egg to produce a hatchling. Despite the technical difficulties, some transgenic lines of chickens have been established by this method. By the time an avian egg outer shell membrane has hardened, the developing embryo blastoderm has two layers consisting of 30,000 to 60,000 cells. In trial experiments, inoculation of the blastoderm stage with replication defective retrovirus vectors containing bacterial marker genes resulted in few chickens on quail carrying these DNA sequences in their germ lines. Although some of these transgenic organisms did not produce virus, they used retrovirus vectors to deliver genes for a product that is to be used as food would ultimately raise questions about its safety, whether real or imagined. Moreover, the size of the transgene that can be introduced into the recipient organism by retrovirus vectors is limited to 8 kb kilobase per, and occasional integration at the initial site is not permanent. Consequently, other methods of transgenesis have been examined. The preferred vehicle for transgenesis of pluripotent cells is that can be maintained continuously in culture and genetically modified by standard methods. To date, only blastoderm cells and the primordial germ cells, which have limited growth in vitro, and stage X, the stage of the embryo in a newly laid egg, chicken embryonic stem cells, which survive for 21 days in the culture, are available for this purpose. Briefly, primordial germ stage 10 embryonic stem of blastoderm cells are removed from a donor chick, transfected with a transgene construct, implanted into the subgerminal space of recipient embryo or freshly laid eggs. At hatching, some of the progeny consisted of a mixture of cells and organisms with non-identical cells from two or more individuals. It's called the chimera. And some of the chicken chimera cells that even were descended from transfected cells become part of the germline tissue and form germ cells. Transgenic lines can then be established from these chimeras by rounds of matings. Generally, the cells of the recipient far outnumber the cells with the transgene. However, the proportion of donor cells can be increased to enhance the probability of obtaining germline chimeras. One strategy entails gamma radiation of the recipient embryos with a dose of 500 to 660 rads for an hour. Before the introduction of the transfected cells, the radiation treatment destroys some, but not all, of the blastoderm cells, thereby increasing the final ratio of donor to recipient cells. The chimeric chicken, despite its inefficiency, this procedure has also been used to produce transgenic chickens. Transgenesis could be used to improve the genetic makeup of existing chickens with respect to built-in viral resistance to viral, bacterial, and coccidial diseases, better feed efficiency, low fat and cholesterol levels in eggs, and better meat quality. Even researchers have also suggested that the egg with its high protein content could be used as a source for pharmaceutical proteins. By analogy to the mammary gland of the livestock, the expression of the transgene in the cells of the reproductive tract of a hen that normally secretes large amounts of ovalbumin could lead to the accumulation of a transgene-derived protein that becomes encased in the egg shell. Ovalbumin constitutes more than 50% of the protein of egg white. Therefore, expression of a transgene under the control of the ovalbumin promoter and regulatory elements can yield high levels of recombinant protein. Yields of up to 1 gram of recombinant protein have been achieved per egg, and considering that a single hen lays more than 300 eggs per year, the productivity of these animals by bioreactors could be substantial. The recombinant protein could either be fractionated from the sterile egg packages or consumed as a nutraceutical. Currently, as a proof of principle experiments, transgenic chickens that synthesize monoclonal antibodies, growth hormone, insulin, human serum albumin, and alpha interferon have been created. Regulatory approval of therapeutic proteins produced in eggs may be more straightforward, as chicken eggs are used to or produce vaccines for injection into humans. Transgenic fish. As transgenic fisheries, as natural fisheries become depleted, production of this worldwide food resource has come to depend more heavily on aquaculture. In this context, enhanced growth rates provide tolerance of environmental stress and resistance to disease are some of the features that may be created by transgenesis. To date, transgenes have been introduced by macroinjection or electroporation of DNA into the fertilized eggs of a number of fish species, including carp, catfish, trout, salmon, Arctic char, and tilapia. The pronuclear fish are not readily seen under a microscope of the fertilization depot. Linearized transgene DNA is macroinjected into the cytoplasm of either fertilized eggs or embryos that have reached the fossil stage of development, unlike mammalian embryogenesis. Fish egg development is external, hence there is no need for an implantation procedure. Development of transgenic fish occurs in temperature-regulated holding tanks. The survival of fish embryos after DNA macroinjection is high, 35 to 80%, and the production of transgenic fish ranges from 10 to 70%. The presence of a transgene is scored by PCR analysis of either nucleated erythrocytes or scaled DNA found in fish are made to establish true breeding transgenic lines. Many of the studies with transgenic fish have been examined with the effect of a growth hormone, transgene on the growth rate. One study of transgene consisting of the upper motor region from the antifreeze protein gene of a fish called the ocean parrot, the growth hormone cDNA from salmon, and the termination polyadenylation signals from the 3' end of the antifreeze protein A gene from the ocean parrot was injected into eggs of Atlantic salmon. This expression system was chosen to enhance the transcription of the growth hormones in cold waters. In general, the transgenic salmon were larger and grew faster than the non-transgenic controls. As all fish construct was assembled to avoid possible biological incompatibilities and that might arise from using a growth hormone gene from non-fish sources. For even greater specificity, an all-salmon growth hormone construct was formulated and microinjected into sockeye salmon eggs. Young transgenic salmon grew much more rapidly than non-transgenic salmon and became adult fish that are on average about 3 to 5 times larger than non-transgenic fish. Theoretically, the faster growth of farmed salmon would lower the cost of the feed and lessen the population of coastal waters in the vicinity of the site of the holding pens. Aquaculture with transgenic fish can be carried out within contained facilities. However, the impact of the accidental release of transgenic fish on natural populations must be considered. In addition to enhancing traits that aid the production of fish or food, transgenesis can be used to generate systems for monitoring aquatic pollutants. One such biosensor system entails the use of transgenic metadata, fish genetically engineered to express red or green fluorescent protein under the control of pollutant response promoters. The metadata is also known as the Japanese killfish, has been a popular aquarium for pet for many years and is currently gaining rapid acceptance as a model organism in which to study biological processes due to its small size, which enables growth in small aquariums, hardiness, rapid development, and transparent body, which facilitates visualization of internal tissues and expression of reported genes such as those encoding fluorescent proteins. Transgenic metadata have been developed as biosensors to detect estrogenic compounds in aquatic environments. Estrogens are sex-steroid hormones that stimulate the development and maintenance of the female reproductive system. And secondary sex characteristics can also regulate some reproductive functions in males. Synthetic derivatives of natural estrogens are used in most contraceptives as a therapy for postmenopausal disorders in women, to treat infertility and endometriosis, and to develop female-only fish populations in aquaculture. A wide variety of industrial chemicals such as bisphenol A and polychlorinated biphenyl species Bs also have estrogenic activity in animals and are used in the manufacture of pharmaceutical, plastics, paints, detergents, and insecticides. Thus, large amounts of estrogenic chemicals are flushed into aquatic ecosystems of domestic aquaculture, actual and industrial wastewater, and sewage and can have a toxic effect on aquatic organ systems. Indeed, one study has implicated high estrogen levels in water wastewater effluent and the feminization of wild male fish that can lead to severe reductions in the fish populations. To develop transgenic Medaka to monitor levels of natural and synthetic estrogens in water, the estrogen responsive promoter from the Medaka vitellogenin gene is cloned upstream of the gene encoding green fluorescent protein and then injected into Medaka eggs. Vitellogenins are normally synthesized in females in response to endogenous estrogens such as 17-beta estradiol. In male response to synthetic estrogenic compounds. Exposure of transgenic fish to 17-beta estradiol, another natural synthetic estrogenic compound, activates the vitellogenin promoter and production of green fluorescent protein that can be rapidly and directly visualized as emission of green fluorescence under normal light in living fish without additional reagents. This system can be expanded into include promoters responsive to either environmental toxins such as heavy metals that control the expression of red and green fluorescent proteins. In some cases, in the same fish, each color will indicate the presence of a different pollutant. Right, going on to the summary part of it. Gene modification of animals by recombinant DNA technology transgenesis entails introducing a clone genes at the genome of a cell that might, after proliferation, embryonic development, and receptor P may be present in the germlines of some of the progeny. These founder animals are then used to establish true breeding transgenic lineages. The clone gene can be introduced into the male pronucleus of a fertilized egg by microinjection delivered into a fertilized egg by using retroviral of transfected embryonic stem cells. Various strategies devised for regulating transgene expression, modifying transgenes and inducing cell death at specified times in particular tissues of a transgenic organism. The KORAH-B recombination system activates transgenes by selectively removing DNA elements that block transcription of inactivated transgenes by excising part of the coding sequence. The TET-on and TET-off protocols use the tetracycline analog doxycycline to either turn on or off the transcription of a transgene. RNAi is often used to decrease knockdown expression of a transgene. These systems have advanced our understanding of gene activity during development and the consequences of either gene overexpression or loss of gene function within a particular tissue. Transgenic mice provide good models for many human diseases such as Alzheimer's disease and are useful to test systems to evaluate potential therapies for human-animal disease. They provide important information about the consequences of defective gene products, the cause of the disease, and the effectiveness of different therapies. Transgenic mice have also been used to produce recombinant therapeutic proteins. The xenomouse, which synthesizes protein human antibodies, was created by transgenesis with a high-capacity YAC carrying almost all of the genes, human and light antibody chains. Genetic manipulation can lead to improved livestock with enhanced growth rates and muscle mass, and increased resistance to common diseases and improved nutrition content of milk and meat products. A major application for transgenic livestock is to use the mammary gland as a bioreactor for the production of protein, pharmaceuticals, and milk. Transgenic animals have been created by DNA microinjection or nuclear cloning. The latter method entails transferring the nuclei from cells that have been genetically manipulated in culture to enucleated oocytes, obtaining with the progeny some animals that carry the transgene in all these cells. Transgenesis of birds, especially chickens, can be used to improve strain attributes. In addition, eggs may be a repository of pharmaceuticals synthesized by transgenic chickens or by delivering recombinant derived medication when an egg is consumed. Genetic augmentation of fish has been directly primarily to improving growth rates and countering resistance to disease. In addition, transgenic fish have been considered as biosensors of environmental pollutants. Transgenes encoding fluorescent proteins under the control of the promoters that are activated fluorescent when particular contaminants such as estrogenic compounds are present have been incorporated into fish for determining the presence of pollutants in water. This lecture ends here in 30 minutes. Time at 30th minute. The review questions will be aired at the end on a later. If possible, I shall do it today. Alright, and on to the review questions. How are transgenic mice created? What is positive and negative selection? How does it work? What are knockout mice? How and why are they established? What are knockout down mice? How are they generated? Describe how the Kree-lock speed recombination system is used to regulate the expression of a transgene. What are the advantages and disadvantages of using transgenic mice as model systems for human diseases? Describe an example of a transgenic mouse that was developed as a model system for a human disease. What is nuclear cloning? Discuss some ways in which transgenic livestock could contribute to human health. Discuss how the mammary gland could be used as a bioreactor for the production of commercial products. Discuss how transgenesis could be used to improve organ transplantation. Describe how a strategy to develop transgenic animals that are protected from infectious disease. Why are pigs carrying a phytase transgene considered to be environmentally friendly? What approaches have been developed to produce transgenic chickens? Discuss how transgenesis might improve fish aquaculture. So I guess that's the end of this chapter on transgenic animals. The next subject, topic on molecular biotechnology and society. We'll be regulating the use of biotechnology and societal issues in biotechnology. So I'll meet you in the next lecture. Take notes, be prepared, make the best use of all the screenshots, and do not commercialize, please. I'll just about recap on the diagrams that you may have missed out. This is one of the Medaka fish, right? Biosensors and environmental pollutants, right? The promoter for the Medaka vitellogen and P gene, P-bit, is cloned upstream of the GFP purported gene encoding fluorescent protein. The rest is written there. Take screenshots and make notes, please. This is the other one, which shows all fish construct used to generate Atlantic salmon with an enhanced growth rate. This is about establishing transgenic chickens by transfection of isolated blastoderm cells. Right? Make screenshots and they should be of help to you. This is about molecular structures of phytic acid, inositol, monophosphate, and inositol. This is about omega-3 and omega-6 fatty acids. Omega-3 fatty acids have long hydrocarbon chains with double bonds between several carbon atoms. All omega-3 fatty acids with double bond is found at the third carbon atom, and details are here. Please make notes. This is a strategy to generate transgenic mice with increased muscle mass. The gene encoding myostatin inactivator, which consists of the N-terminal propeptide domain of myostatin, was introduced in Y chromosome mice, embryonic stem cells, under using two-step procedure. Please take notes and take screenshots. This is about the myostatin that consists of an N-terminal propeptide domain LAP and an active C-terminal domain. That's it about this chapter. Make notes. Best wishes. Thank you. Bye.